So for this video, we're going to be discussing DNA structure and replication. In class, we talked about some of the scientists and experiments that they did to help us understand that DNA is the genetic material. So in class, we discussed some of the scientists and the experiments that helped us understand that DNA is a genetic material. We're going to start here with Watson and Crick. So these are the two most famous scientists that are credited for correctly describing the structure of DNA. In 1953, they had an article in Nature magazine printed, and in their article, they discussed the structure of DNA. And here's a picture. I believe this picture on the left side is from Time magazine. This is James Watson at the very bottom, and then this is Crick at the top there. At the time when Watson actually Watson discovered the structure of DNA, he was only 25 years old, so he was really young. And at that time, in the 1950s, he was, from what I've heard, back in the 1950s, he was a bit of a celebrity and a very eligible bachelor, because he was in his mid-20s and late 20s when he was recognized for his discovery of DNA, which was one of the most biggest discoveries of that time. So what did they propose? They proposed that DNA is made up of two strands. So that's what we mean by double. The double refers to the helix, the two strands of DNA, and then the helix refers to the structure of DNA being like a twisted ladder, okay? So you have two strands. Here is one strand here. Okay, one strand here, and then here's another strand. And so it's like a ladder because the nitrogenous bases that they proposed are holding the two strands together. Okay, so like a ladder structure. Okay, and they said that the two nucleotides make up the rungs of the ladder, like the thymine and adenine, the guanine, the cytosine, guanine, adenine, thymine. Okay. And then also we're going to be talking about, there's a little quote at the very bottom. We're going to be talking later on about how DNA replicates. So they propose a structure of DNA, and from the structure of DNA, they were able to figure out how DNA actually copies itself, how it actually replicates during the S phase of the cell cycle. Okay, and you can see here that one strand of DNA, if we just focus on one strand, here is a nucleotide. Here is another nucleotide. Here's another nucleotide. So the monomers of DNA, remember that term monomers when we were going over biochemistry? So the monomers, the parts that make up DNA, are called nucleotides, okay? Nucleotides. And nucleotides are made up of three parts. You have the phosphate group, the deoxyribose sugar, and then the nitrogenous base. Why do we call it nitrogenous base? Is because when we look at the structure of the nitrogenous base, there's nitrogen in there, there's carbons in there, there's hydrogens, okay? So remember, DNA is a type of organic compound. It belongs to the nucleic acid organic compound group because there's carbons in there, okay? And the nitrogenous base contains nitrogen, carbons, hydrogens, and oxygens. And then the deoxyribose sugar has the carbons and oxygens and hydrogens. And then the phosphate group here, you can see it. It's a phosphate atom surrounded by oxygens and a hydrogen, okay? All right, moving on to the next slide here. So let me just move my head out of the way so you guys can actually see this nucleotide. We're going to be going ahead and labeling the different carbons that make up this deoxyribose sugar. This should actually say deoxyribose because DNA is deoxyribonucleic acid, whereas RNA is ribonucleic acid made with ribose. Okay, so I'm going to just go ahead and do that. Deoxyribose, okay? So here's our nitrogenous base. There's a carbon here in that corner of our deoxyribose sugar. There's another carbon in this corner. Another carbon, and then this is the next carbon. Okay, so this is a pentose sugar. Pentose, right? Maybe I'll write it up here. Pentose sugar, because it has 5 carbons. Okay, it has 5 carbons. So here we have carbon number 1, carbon number 2, carbon number 3, carbon number 4, and carbon number 5. And you do need to know the number of the carbons, or the number carbons, because that's going to tell us the directionality of DNA. Remember how we said that DNA is made up of 2 strands? Well, each strand is going in a specific direction, okay? And when we say direction, we're going to be talking about like a 5' end and a 3' end. It's sort of like a train. There's a front and a back, okay? So think of like each strand of DNA being like a train where there's a front part and then a back part, okay? Oh, and my face is covering up Fillmore, so let me go ahead and move my face over here then. So yeah, this is going to be important when we talk about the directionality of DNA, the carbons. So DNA, the backbone, what do we mean by the backbone of DNA? Remember how I said that DNA is like a ladder, right? The backbone, the sides of the ladder is what we mean by the backbone. And the sides of the ladder, you have the phosphate groups, sugar, phosphate group, sugar. So the phosphate group sugar, that's the sides of the ladder, whereas the nitrogenous base are the rungs of the ladder. Okay, the rungs of the ladder. So DNA has a 5' end and a 3' end. The nucleotides in DNA backbone are bonded from phosphate to sugar between the 3' and the 5' carbons. Okay, so when you look at, let's take a look at this right over here, this nucleotide right here. This would be the 3' carbon. This would be the 5' carbon. Okay, so the 5' carbon is attached to its own phosphate group. That phosphate group is attached to the nucleotide above it, or in front of it, at the 3' carbon of that other nucleotide. Okay, so this other nucleotide, it's attached to the phosphate group at the 3' carbon. And then its phosphate group is attached to its 5' carbon. Okay, so that's what makes up the backbone. Okay, and so we say that DNA has, like if this was a very short train just made up of 2 nucleotides or 2 boxcars. Okay, this is the 5' end, this is the 3' end. Okay, the reason why it's called the 5' end on this side and the 3' end on that side is because which carbon in the deoxyribose sugar is closest to that end? It's the 5' the 5' carbon is the closest to that end. The 3' carbon for that deoxyribose is down here. So the one at the far end, okay, and then if you look over here on the 3' end, the closest carbon in the deoxyribose sugar is 3'. Okay, so this is the 3' end, that is the 5' end. Okay? So yeah, and you can see that again in this diagram. Okay, this is the 5' end, this is the 3' end. So this is one strand of DNA. This is the 5' carbon attached to its own phosphate group is closest to that end. And then when you look at this deoxyribose, this is carbon number 1, carbon number 2, carbon number 3, carbon number 4, carbon number 5. And the closest carbon to that end would be the 3. Okay? So this is one strand of DNA right over here. There's another strand of DNA over there. And the other strand of DNA is going in the opposite direction. Remember how I said directionality of DNA, okay? Well, the other end, this is the 5' end, this is the 3' end, okay? So we say that DNA is anti-parallel. The 2 strands of DNA are anti-parallel. Anti means opposite. It refers to the primes. The prime ends are opposite. But then parallel means that they're going side by side, okay? So the 2 strands of DNA are parallel to each other, but the directionality of the strands are opposite to each other. This is the 5' to 3' end. This is the 5' to 3' end. They're going in opposite directions, okay? So this is the 5' end, this is the 3' end. This is the 5' end. This is the 3' end. Okay? So the complementary strands, when we say complementary, remember how A goes with T, adenine will pair up with thymine, guanine and cytosine pair up, guanine and cytosine pair up, thymine and adenine pair up. So the complementary strands run in opposite directions. Okay? And so let's talk about bonds that are holding that DNA molecule together. Between the phosphate groups and the carbon are very strong bonds. Okay, they're strong bonds called covalent bonds. Remember, with covalent bonds, they're sharing electrons, and when they're sharing electrons, the energy levels are overlapping, and they're sharing electrons, and so they would be very strong bonds. And there's a phosphodiester covalent bond there, a phosphodiester covalent bond there, and then there's one right there. And maybe I'll just go ahead and just draw them all, like point at all of them. So there's one right there, one right there. Okay, so it's very strong bonds. So it would be difficult to cut DNA, going in that horizontal direction. However, DNA is held, the two strands are held together by very weak hydrogen bonds. You guys learned that hydrogen bonds are pretty weak, right? Remember, those are the bonds that hold water molecules together. Like when you jump into a swimming pool, you're bursting through you're breaking all those hydrogen bonds. I mean, we can't walk on, the water or like rest on the water like a leaf would float on the water, or how a water strider could walk over the hydrogen bonds. We can't do that, okay, because we're too heavy. We would break through those bonds. They're very weak bonds, okay? So between thymine and adenine, there's 2 hydrogen bonds. Between guanine and cytosine, there's 3 hydrogen bonds, okay? And you can see that there. Before Watson and Crick proposed their structure of DNA, some scientists thought that actually there were 3 nitrogenous bases that made up the rungs of this ladder, or this 3-dimensional structure of DNA. So their hypothesis was that there's actually 2 nitrogenous bases that make up the width, okay, the width of the ladder, the width of a DNA molecule. So with this idea of hydrogen bonds, knowing this, that there's weak hydrogen bonds that hold the 2 strands together right down the middle, we're going to be talking about how DNA replicates. Watson and Crick also proposed that the hydrogen bonds between the nitrogenous bases enable DNA to easily copy itself, okay? So we're going to be talking about the mechanisms of DNA replication as well. So base pairing in DNA, you have adenine and thymine, guanine and cytosine, and we said that between adenine and thymine there are 2 hydrogen bonds. Between guanine and cytosine there are 3. Okay, adenine and guanine are known to be, they're just called purines. Purines are the ones with the double rings. Do you guys see that? This is 1 carbon ring. This is a 2nd carbon ring. 1 carbon ring, 2 carbon rings. So the purines have double rings, whereas the pyrimidines are single rings. Okay, single carbon rings with nitrogens in there. Do you guys see? Oh, they're called nitrogenous bases because you can see that, right? Nitrogen, 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 etc. So the purines are the adenine, guanine, which have double rings. Pyrimidines are thymine, cytosine, which are single ring. Okay, and A goes with T. There's 2 hydrogen bonds. Guanine goes with cytosine. There's 3 hydrogen bonds. So, I mean, between guanine and cytosine, it is a stronger bond only because there's 1 more hydrogen bond. But again, hydrogen bonds are pretty weak, okay? So that brings us to copying DNA. How does DNA replicate or copy itself during the S phase of the cell cycle? Well, Watson and Crick, they proposed that since the hydrogen bonds are holding the 2 strands together very weakly, perhaps the 2 strands can come apart, pull apart, and then each one can act as a template. And if they act as a template, they can act as a template to make a complementary strand. Because remember, adenine pairs up with thymine. guanine pairs up with cytosine, and you're going to end up with 2 copies if the original 2 strands, when they separate, they act as templates to make a complementary strand. Okay, so for example, if you look down here, this is the 2 strands of DNA, and then they're going to separate, and then each strand is going to act as a template. So on the left side we have A, C, T, A, G, and A is going to pair up with T, okay? And then that C is going to pair it with G, and that T is going to pair it with A, that T is going to pair it with T, G is going to pair it with C. So you guys can see, okay, that now, as the original strands act as templates, new nucleotides can come in and they can pair up. They can pair up according to complementary base pairing rules. A goes with T, C goes with G, okay? This is what they call the semi-conservative model, okay? I wonder why it would be called semi-conservative. Well, semi means like partly, right? And conservative means to maintain. So when you look at the 2 new strands of DNA, part of half of each new strand is old, okay? It's the original strand. So that's what we mean by semi-conservative. Part of it is the original strand, okay? Part of the DNA molecule is original. So conservative means to like keep, maintain. So semi-conservative. There were other I guess hypotheses as to how DNA replicates, but I think we're going to go over that at the end, okay? So if we have DNA replicating, in human cells we have 46 linear strands of DNA, okay? So 46 strands of DNA, and I'm not going to like draw all 46, but think about, that's a lot of linear pieces of DNA to be copied. And instead of the DNA splitting from one end and then going down and each strand acting as a template, there's actually what we call replication bubbles, where the DNA will split somewhere in the middle of the linear strand of DNA and then come apart. Okay, it's sort of like, if you have a broken zipper, it's broken in the middle and then you pull it apart. That's what's happening in the replication bubble, or the origin of replication. So the original strands are like separating, okay? And then each original strand in dark blue here is going to act as a template for new nucleotides that are going to join up by complementary base pairing. And you can see that in the light blue color. And that is called the daughter strand. So the semi-conservative model, you can see at the end, you have an old strand, new strand, old strand, new strand. So you end up with 2 pieces of linear DNA, and it's semi-conservative because one strand is the template, came from the original strand of DNA, and then the other opposite strand is new, okay? All right, so let's go ahead and talk about DNA replication. Well, the first step is to unzip. Okay, it's to unzip or to unwind. And you guys, when we talk about DNA replication, there's a bunch of enzymes involved with replication. There's a lot involved. And you do need to know what each enzyme does, okay? The first step is helicase. Helicase binds to the DNA helix. That's the first step. It's going to bind to the DNA helix and unwinds that part of the DNA helix, okay? So you can see helicase here. It is unzipping the DNA in this direction and then unzipping it in this direction. They're going in, there's 2 helicase molecules or proteins or enzymes, and they're going in opposite directions, and they're unzipping it going in opposite ways. And then each strand, see how now they're unpaired? The hydrogen bonds are now broken between these 2 strands. Now each one's going to act as a template for a new DNA to be made. Okay. Oh, that was a little scary, huh? So yeah, this is that's Fillmore, and he's just saying a pickup line. Okay, pickup line. I'd love to be helicase and unzip your genes. That's a little racy, huh? Okay. Oh, before I move on to the next slide, I did want to mention, and these proteins are not that important, but no, not important for, you know, like tests and quizzes, but they are important in DNA replication because they play a very important role. They're single-stranded binding proteins, these little turquoise guys. They basically hold the DNA strands stable as helicase unzips or unwinds or takes the DNA strands apart. Because if you think about it, like if you have yarn, I might have yarn somewhere, and I should have had it with me. If I have yarn and I'm pulling the two pieces of the yarn apart, you know, and if I'm pulling them apart really quickly, they could get tangled, right? They get bunched up, and then they get tangled. So the single-stranded binding proteins helps prevent the tangling, the DNA from actually tangling as the helicase unzips them, okay? So, you know what? Let me just really quickly describe what Helicase is doing, maybe with a white screen here. Okay. So if I have, so this is my original two strands of, that's so not straight, but it's okay. Two strands of DNA. Okay, that's my original one. Then Helicase is going to come in right over here and maybe over here, and both of them are going to start unzipping, going in opposite directions. Okay, so you're going to have this bubble that's starting to form. And so I should have probably drawn it a little bit longer on both sides. Okay, excuse me. So then I have helicase going in this direction. What they're producing are forks, okay? Like it's called a replication fork. You know, like when you meet a fork in the road, you can go in either direction. So it's sort of like a replication fork is being made because they're now like two roads, okay? Because they're separated now. So now you have the rungs of the letter are broken. Those nitrogenous bases are no longer paired with each other. Let's say like this was A, and then this is T, and this was G, and this was C. They're no longer together. They're separated. And so these are now templates for new nucleotides to come in and join up. And then eventually, as helicase makes its way down, unzipping, you're going to have the two strands that are going to act as templates for complementary base pairing. Okay, and I've drawn them separated, but in actuality, there's another enzyme that's going to jump right in that replication bubble and start adding new nucleotides. But just for simplicity's sake, I've just drawn the two strands separated because that's what helicase does. Helicase is going to unzip the DNA, unzip the genes, okay? So the second step is just building a daughter strand, okay, a daughter strand, adding new complementary bases. And the enzyme that does this is called DNA polymerase 3, okay? DNA polymerase 3. There's three different kinds of DNA polymerase. Polymerase 1, polymerase 2, and polymerase 3. DNA replication uses DNA polymerase 3 for adding new nucleotides that are complementary to the template strand, okay? So let's say that this is our DNA polymerase 3, and it just goes along the template and starts adding new nucleotides that are complementary. Okay, so Fillmore is asking a good question. We're missing something. What is it? Okay, I mean, honestly, were we really thinking about it? I don't know. But we're missing the energy for bonding. Do you guys remember in the beginning of the year we talked about how every chemical reaction requires an input of energy. Remember what that's called? Activation energy. Activation energy, yay! Okay, so you guys remember activation energy. And so activation energy is the amount of energy that's needed in order for a reaction to happen. Enzymes will lower the activation energy so that reactions can happen. And these are technically chemical reactions because bonds are forming. Okay, bonds are forming. So DNA polymerase is the enzyme that brings them together, okay? But what gets the nucleotides there in the first place are actually nucleosides. I've been saying nucleotides, okay? Like this is a nucleotide, right there. But a nucleoside actually has 3 phosphate groups. For example, ATP. Remember ATP? It's the energy providing molecule. Okay, there's energy that's held in these bonds between the phosphate groups. Remember, energy is released when that last phosphate group, that inorganic phosphate, when it's released, energy is released, and it forms, goes from ATP to ADP. You remember ATP? Are there other ways to get energy out of it? Yes, there is, okay? You can actually break off the other phosphate group and energy is released. So actually, you can get AMP, okay? You can get AMP. So ADP was adenosine diphosphate with the 2 phosphate groups. AMP is adenosine monophosphate, mono meaning 1, okay? So energy can be released with when the bond between the first and the second phosphate group is broken. So the nucleosides, they're called nucleosides, they come with their own energy. So when DNA polymerase 3 joins up the complementary base pairs, the nucleotides, they provide the energy that's going to help build the complementary strand, okay? So yeah, there's GTP. It's not just ATP. There's GTP, TTP. I love these sound effects. There's CTP. Okay, you know, we did talk about that in class, how there are other nucleotides, right, that can provide energy. It just so happens that ATP is the most utilized in a cell as a source of energy. But there are, you know, they could use GTP. Actually, you know what? We did, I think it was in cellular respiration in the Krebs cycle, where we did talk about GTP. Some GTP, I think we did, okay? So the nucleotides or nucleosides with the 3, okay? They have 3 phosphate groups. They come with their own energy, and the enzyme is what brings them together, the complementary base pairing, okay? So here we go. Oh, and another thing is that DNA polymerase 3, it can only add new nucleotides in one direction. Okay? It's only going to add at the 3' end. So do you guys see, this is the template over here on that side. And the template this is basically the reading strand, what's being read, it's the template. On the template, this is the 3' end, and down here, that's the 5' end. And then the strand that's being made, this is the 5' end. And then this will be the 3' end down here. How do we know that? This is the 5' carbon. 5' carbon is closest to that end. This is the 3' carbon. As I was saying, DNA polymerase can only add to the 3' end. So the next nucleotide will be added here, like so. Oh my goodness, I don't know if I can draw a proper sugar. This is so bad. But yeah, let's just pretend it looks pretty, okay? So there's my nucleotide. It attaches at the 3' end, the 3' sugar, 3' carbon of the sugar, okay? So DNA polymerase can only add to that carbon. So there's a limitation, and there's the 2 inorganic phosphates that come off. There's thymine. Okay, and then there's cytosine. Okay? So that's one of the limitations with DNA polymerase, is that I can only add to the 3' end. And it's going to cause some difficulty when we are dealing with the replication forks. Remember when Helicase, it unzips and goes in opposite directions? Okay? Because DNA, the original molecule has a 3' end and a 5' end. Okay? So as a result, it can only add to the 3' end. So here's that replication fork. There's Helicase unzipping the DNA. Okay? This is the 3' end of the template. Okay, this is the 5' end. Let me move my head. I gotta move. Sometimes I forget that you can't see beyond my head. Okay, so this is the 5' end of the growing strand. The growing strand, and in this case, this is called the leading strand. This is the strand that's being made by DNA polymerase, and this is the 5' end, and it can add the DNA polymerase can add continuously to the 3' carbon of that deoxyribose sugar. So it's called a leading strand because it's just leading on, being made smoothly, going as the replication fork is growing in that direction. The leading strand just continuously adds more and more to the 3' end. The problem on the other template, see the other strand that's a template, the problem with that one is that this is the 5' end, okay? And then therefore, remember how we said DNA is anti-parallel? If this is the 5' end and this is the 3' end, then the opposite strand, this is the 5' end, this is the 3' end. What does that mean? That just means that for the growing strand of DNA, it has to be added. The nucleotides have to be added to the 3' carbon, okay? So you can't add it going in this direction, you have to add it going in that direction. So like as the replication fork is opening, it starts here, okay? And then it adds. And then it starts here and it goes and adds. And as the replication just keeps on opening up, it starts a new strand and it continues on and it keeps on building in that direction. It's disjointed, it's not continuous like the leading strand, but you end up with these little fragments. So you guys, the next fragment is going to be over here, right? This is going to be the next fragment that's being made. As the replication fork opens up, it's growing and it's opening it up. There's more template being shown, and then you keep on adding a new fragment, okay? And these are called Okazaki fragments. I love saying that, Okazaki fragments. There's a Japanese scientist that discovered these fragments. And so you end up with all these little pieces of DNA or fragments, and then they have to be glued together using another enzyme called ligase. Okay, and that's why it looks, ligase is drawn like this, because it's like glue, liquid glue. Okay, so let me show you. I think there's a good enzyme, not enzyme. It is a good enzyme. Ligase is a good enzyme. Okay. Oh, yeah. Okay, so this is a good diagram is what I wanted to say. So here you have the DNA original strand, 3' end, 5' end on that side, 5' end, 3' end on the other side. They're anti-parallel, okay? So you have two replication forks, one going in this direction and then another going in this direction, okay? So this is where it started right here at this point. Okay, and as it starts at this point, this is the 5' end of the new strand, because this is the 3' end. Okay, so this is the 5' end, and it's you just keep on adding to the 3' end, no problem. That's why it's called the leading strand, because it's nice and continuous. DNA is being added continuously to the 3' end as the growing replication fork goes in this direction. And as the growing replication fork continues in this direction though, okay, as it's opening up, 5' end is over here, and at the 3' end, nucleotides have to be added. So just imagine the replication fork opening up even further, then a new 5' end here, and then you get another fragment, okay? I apologize, I feel like this pen doesn't work very well. But if this was the point where it opened up the replication bubble, then right over here was the 5' end to start with. Then this side you have the leading strand going in this direction, okay? And then as the replication fork goes in this direction, the leading strand can continue to add complementary strands to the template, okay? But then opposite to that, as the replication fork is opening up, you have to have fragments. You have to have those Okazaki fragments. So the reason why we have Okazaki fragments is because DNA polymerase can only add to the 3' end, and when a replication fork is opening, remember the DNA, the templates, there's two strands. One is 3 to 5 there, and then this one's 5 to 3. And so DNA can only be added to a 3' end. Okay? So, yeah, so you have leading and lagging strands, and then the lagging strands are called Okazaki fragments, and they need to be glued together with ligase enzyme. So that's the thing about DNA polymerase 3. Okay, so this is our growing replication fork as it's unzipping by helicase. Helicase is not drawn here, just for simplicity's sake. As the helicase is unzipping the DNA, the leading strand can continuously add at the 3' end, but then the Okazaki fragments, as the replication fork opens, a new 5' end starts here, and then you have another fragment being made, okay? However, in order, I don't know if you guys can see that down there, because sometimes whenever I do a screen record, you can't see the stuff at the bottom. I totally forgot about that. Okay, well, I'll try my best to just like read whatever's at the top and at the bottom. So this is starting DNA synthesis, RNA primers. So here's another thing about DNA polymerase 3. I said that it's limited in that it can only add to a 3' end. It can only add to a pre-existing 3' end of a sugar, okay? And so there's another enzyme in DNA synthesis called primase, okay, primase. And primase is what starts off the leading and lagging strands. So we haven't talked about primase yet. We just said, oh, DNA polymerase can add to the 3' end of a DNA strand. But to start it off, you need a primer, okay, a primer. A primer is a little piece of RNA. So here's a primer. I'm circling all the primers. Primers are pieces of RNA, okay, pieces of RNA. And they have, just like DNA, they have a pentose sugar. So I'll just draw it like this, okay? This is carbon number 1, carbon number 2, carbon number 3, carbon number 4, carbon number 5, okay? So this primer has a 3' end as well, okay? So it starts off, DNA polymerase 3 is going to add to that primer. It's going to add to the RNA, a little primer RNA, okay? And the enzyme that puts down the primer is called primase. So primase is the enzyme, like this guy in orange, puts down the primer, then DNA polymerase can start adding to the 3' end. So just like, I guess primers, when you like do painting, primers is you set it down, it's like the base, you set it down, and then you can paint over it, right? So think of it that way, DNA polymerase 3, it needs a primer to start doing what it does, which is to add complementary base pairs. Okay, so the primase starts it off by adding a primer, a small RNA sequence, and then DNA polymerase 3 can start adding to the 3' end. But you guys know that your DNA is DNA. There's no RNA pieces. There shouldn't be RNA pieces in your genome, okay? So there's another enzyme that's going to replace the primers with DNA. So it says it up there, replacing RNA primers with DNA. And that would be DNA polymerase 1, okay? So DNA polymerase 3 adds new complementary DNA nucleotides to the template, but our DNA polymerase 1 is going to come along and just replace the primers with DNA nucleotides. It's going to go in there and remove the primers and replace it with DNA, okay? And then the ligase comes in, just like with the Okazaki. Well, this is the fragments, actually. These are fragments right over here. This fragment, once the fragments, the primers are replaced with DNA, then ligase comes in and glues the fragments together, okay? Here, though, the problem is that there's no nucleotide over here, okay? So you can't replace that RNA with DNA because there's no other nucleotide in the front to add the first nucleotide to. Does that make sense? So there's nothing in the front where we can actually add another DNA nucleotide. So as a result, DNA polymerase will come in, remove the RNA, but then DNA polymerase 1 cannot add to that. So as a result, DNA a piece of DNA will be missing. Okay, DNA will be missing. And as DNA synthesis occurs in a cell over and over again, like as that cell line is replicating over and over again, the DNA is eventually going to get shorter because now this strand of DNA is going to act as a template for the next time that it replicates. Let me just erase this mess here, okay? So if you look at this strand, if this is, let's just pretend that this is a strand of DNA, a new daughter strand of DNA. Now this is going to act as a template. And that template is going to have a piece of the DNA missing. And then when it, when the DNA strands are going to act as templates for new strands of DNA, every time there's going to be a piece of DNA missing because it cannot be replaced. The RNA in the primers cannot be replaced. So that brings us to this discussion that chromosomes will erode, chromosome erosion, because every time DNA is copied, that primer to begin with, at the end of that DNA, cannot be replaced. So all DNA polymerase can only add to the 3' end of an existing DNA strand. There's going to be a loss of bases at the 5' ends. That's the 5' end. Okay, and chromosomes get shorter with each replication, and therefore it's going to limit the number of times that the cell can actually divide, because what's going to happen is that eventually as the DNA gets shorter and shorter and shorter, there's genes, right? There's genes on the DNA. And the genes are going to start getting deleted, right? The genes will be deleted, and then when genes are deleted, that can lead to mutations, and then the cell could be, it can be destroyed by the immune system, destroyed by the cell, okay, because apoptosis, right, cell suicide, because there's a mutation, okay? So this is a really cool evolutionary adaptation, is that on all of our chromosomes we have telomeres, okay? Telomeres are non-coding sequences at the end of DNA. What I mean by non-coding is that they do not code for protein. So they are no protein. They don't code for proteins. Basically, guys, they're not genes. Okay, they're not genes. They're just A, T, G's, and C's, the adenines, thymines, guanines, and cytosine, that don't actually code for anything. So basically, as the DNA gets shorter and shorter, you can afford to lose that piece of DNA because it doesn't code for anything important. It's sort of like buffer. Like you have enough space to just, like, as the DNA gets shorter and shorter, you can sacrifice it, okay? Because there's nothing important there. I wish I had an analogy for that, but I don't. It's just a buffer zone, okay, where you have a lot of DNA that doesn't code for anything. And those are called telomeres. And you have them at the end of every DNA strand. At the end of every chromosome, you guys have telomeres. It's buffer zone, because as DNA replicates, it gets shorter and shorter. There is, however, an enzyme called telomerase that can add telomeres to the end of our DNA. And cancer cells have very active telomerase. Remember we talked about HeLa, right? The HeLa cells, the immortal life of Henrietta Lacks. She's immortal. Her cancerous cervical cells are immortal because she had very active telomerase enzymes that keep on adding to the ends of the DNA. As a result, as a result, her DNA is not getting shorter, genes are not being compromised, genes are not being mutated, or genes are not being deleted, okay? So cancer cells have a lot of telomerase activity, whereas a mature adult probably would not. It says there at the very bottom, so stem cells and cancer cells have high telomerase activity. Okay, because stem cells are baby cells, and they're going to develop into mature cells. And so because they're baby cells, they have a lot of telomerase to, like, keep the cell dividing and growing for as long as it can before it matures. So again, here's just another diagram showing you what's happening at the replication fork. Helicase, there's helicase, splitting the DNA strands. It's unzipping the DNA strands. This is the replication fork, okay? And on the original strand of DNA, this is the 5' end, this is the 3' end. Opposite to the 3' end is this 5' end, and DNA polymerase 3 has been adding. It's been adding new nucleotides at the 3' end. So this is the leading strand because it's a strand of DNA that's just being made continuously. Whereas on the opposite side, maybe the replication fork started over, started somewhere over here, okay? And so primer was set down. Primase set down a primer, and then from there, DNA polymerase was adding to the 3' end. So this is the 5' end, this is the 3' end, okay? This is the 5' end, this is the 3' end. So you have primase setting down a primer, and then DNA polymerase 3 can add to the 3' end. And then next comes DNA polymerase 1. It's going to remove that primer, or actually these are the primers in pink, and replace it with DNA nucleotides. After all the primers are replaced with DNA nucleotides, then ligase is going to come in and glue those fragments together. Okay? Oh, and it's kind of interesting that the two scientists that discovered DNA polymerases 1 and 3 are father and son, Arthur Kornberg and Thomas Kornberg. Arthur was Thomas's dad, and Arthur Kornberg discovered DNA polymerase 1, and then Thomas discovered DNA polymerase 3. Okay? DNA polymerase 2, some of you guys may be wondering, what does DNA polymerase 2 do? There's still controversy as to what it does, but we know that it exists in prokaryotic cells. So DNA polymerase 2 is found in bacteria, in prokaryotic cells, and we believe that it plays a role in DNA synthesis for prokaryotes. It's sort of like a backup of DNA polymerase 3. So bacteria have DNA polymerase 3 as well, and DNA polymerase 2 supposedly is like a backup of it. Okay? So DNA polymerase 3, it's like the main DNA builder, right? It's adding complementary nucleotides to the template strand. And then DNA polymerase 1 is the one that comes in and is going to remove the primer. That's what we've talked about so far. It removes the primer, replaces it with DNA nucleotides. It's also responsible for editing and repairing any mistakes, okay? If there's any mistakes that are done by DNA polymerase 3, so you know how DNA polymerase 3, puts a complementary strand there. If it makes a mistake, DNA polymerase 1 is the one that will actually fix the mistake, okay? I guess it's an editor, okay? So yeah, DNA polymerase 1, it proofreads and corrects typos, repairs mismatched bases, removes abnormal bases, repairs damages throughout your life. It reduces error rate from 1 in 10,000 to 1 in 100 million bases. That's very good. That's a very good thing that we have DNA polymerase 1 to do that. Okay, so let's say that, in this picture here, this molecular structure of thymine is that it can form a dimer, meaning like it can bond with a neighboring thymine. So if this is thymine and thymine together side by side on the strand, they could actually form a covalent bond. And when they form a covalent bond, that's not good because it causes the DNA to fold, or bend, I should say, and when it bends, then the thymines cannot form hydrogen bonds with the complementary adenines opposite to it. And so what's going to happen is that a nuclease enzyme is going to cut the damaged ones and then repair it. DNA polymerase 1 will come in and repair the damage, okay? So I haven't mentioned nucleases, but there are nucleases in our cells that go around and like cut up nucleotides. They break the phosphodiester bonds between the nucleotides in the, what do you call it? The backbone, the sugar phosphate backbone. Nucleases do that. They cut the sugar phosphate backbone, the phosphodiester bonds. So, like, how fast does this happen? How fast does DNA replication happen? With E. coli, it's less than an hour. In some cases, it's like 20 minutes, okay? And you know what? The thing is with E. coli is that they are very simple organisms. They don't have as many base pairs as we do. They only have 5 million base pairs, and so they have less DNA to copy. So it only takes like 20 minutes. Whereas a human cell, there's 6 billion base pairs, and so it's going to take a few hours. Maybe 24 hours, so a few hours to 24 hours. It's remarkably accurate because of DNA polymerase 1. Only 1 in 100 million base pairs is there a mistake. Okay, so it's around 30 errors per cell cycle. And you might be thinking, Ms. Shem, 30 errors per cell division? That sounds kind of like a lot. I mean, can we afford to make that many mistakes? Well, the thing is that, I think it's like 97% of your DNA actually does not contain genes. Okay, only 3% of your DNA, when you look at the genome, only 3% of it actually codes for proteins or make up genes. Okay, the other 97%, they could either be telomeres. Okay, telomeres. This is a different chapter, but we'll talk about what the 97% of our DNA does. But a lot of that 97% play important roles. Okay, they might be part of regulating which genes are going to be expressed. So they play an important role in what genes are going to be expressed. Some of them, are what we call pseudogenes. They used to code for genes, but there's mutations in them that now make them non-functional. Okay. So what does DNA replication look like? This is a microscopic photo of DNA. Here's a replication fork there. And so here's, one strand, daughter strand, there's another daughter strand, there's another replication fork. So actually this is a drawing of what's going on. Replication fork, replication fork, replication fork. So I don't know what cell this is in. It might be a human cell, okay? A picture of replication forks all throughout the genome. So as you guys know, this PowerPoint is based on Miss Foglia, and she put this in here. She was so funny. So back in the old days, back when I was like high school, college, we didn't have dating apps. Hopefully you don't have dating apps on your phone, okay? But you guys have heard of dating apps, right? You don't have dating apps, but you have heard of them because you're too young to be actually having them on your phone. But before dating apps was invented, if you were like kind of in need, if you were searching for a significant other, you had to advertise it in the newspaper. So here is a clip from the newspaper of some person, I think, gentleman, who was looking for a specific type of female, okay? It says here, I'm an athletic, mature, independent, 33 years old male with a youthful appearance of 23. I'm already suspicious. It's already sus, right? I'm a single white male and a working professional in the research industry. I'm 5'10" tall, 175 pounds, with light brown hair, hazel eyes. I'm a non-drinker and non-smoker. I've never been married and have no dependents. Currently seeking female companion who enjoys the outdoors and understands DNA replication. If interested to find out more, call box 1044. Okay, so funny because this is an actual advertisement, an actual ad in the newspaper. Some guy is looking for a female that understands DNA replication. So obviously he's looking for a specific type of girl, huh? Okay, let's do some review questions, and then I'm actually going to go back to a few slides in the PowerPoint and talk about the experiment that proved that DNA replicates in a semi-conservative manner. Base your answers in the following questions on the choices above, or below, sorry. Oh my gosh, not above. Okay, so we have answer choice helicase, polymerase, ligase, primase. So number 1 brings together the Okazaki fragments. So which of those enzymes bring together the Okazaki fragments? Yes, you're correct. Ligase. Okay. Number 2, adds nucleotides to the leading strand. Yes, polymerase, and specifically it's polymerase 3. Okay. If I had another answer choice 4 that said corrects any errors in DNA replication, then you would say DNA polymerase 1. Okay. Okay. Number 3, recognizes the origin of replication in the DNA. So which enzyme is going to remember, the origin of replication is where the replication forks start. Okay, so do you guys remember which enzyme produces the replication forks? Helicase, okay? And then after helicase unzips, it's the primase that comes in next, right? So helicase is number 1, primase is number 2, setting down a primer. DNA polymerase 3 is the next enzyme that comes into play because it's going to start to add complementary nucleotides to the template strand. And then ligase is the last enzyme, right? Ligase enzyme is the last enzyme to come in and glue all the Okazaki fragments together after DNA polymerase 1 replaces the primers with DNA nucleotides. So there's a lot of enzymes, you guys. There's a lot of enzymes involved with DNA replication. There's actually 5, right? Helicase, polymerase 3, polymerase 1, ligase, and primase. In DNA synthesis, DNA is read 3 to 5 and made 5 to 3, or read 3 to 5, made 3 to 5. Okay, so when I say read, that is the template strand. Which template strand is being read? And then it's made. I think that, remember we said the DNA is made from 5 to 3. DNA polymerase can only add to the 3' end. So it starts at the 5' end, but it adds to the 3' end, okay? So the answer is A, because the opposite strand, so let me just to draw it. So this is the 3 to 5'. Yeah, so this is the template, okay? And then this is the 5' end of the new complementary strand, and then this would be the 3' end, and it's just adding. DNA polymerase 3 is just adding to the 3' end. So the 3' end is just getting longer and longer, okay? So this would be like the leading strand because it's a smooth, continuous strand that's being made and adding new nucleotides to the 3' end. Okay? Oh, and then, so if this is our primer, can it be replaced? No, it cannot. So once DNA polymerase 1 comes in and removes the primer, it cannot add DNA to that section. And so that this would be the 5' end. This is the new 5' end. It's shorter. The DNA strand is shorter. Okay? All right, let me just go back really quickly to the slide that I want to go to regarding the experiment that proved that DNA replicates semi-conservatively. So let's actually start over here with this slide, okay? So, there were 3 different theories as to how DNA replicates, and Watson and Crick, they were the ones that proposed a semi-conservative model. But there were 3 hypotheses, like conservative, semi-conservative, and dispersive. Conservative means to like completely maintain, okay? So here's the 2 strands of the original DNA molecule. And the theory was that it would unzip and then both would act as templates. But after the templates were used up to form 2 new strands of DNA, the template strands would come back together. And then the 2 daughter strands would come together. Okay? And then after several generations or 2 generations of DNA being replicated, there's the original. The original is conserved. Oops, let me go back. The original is conserved, and then you have new daughter DNA, new daughter DNA, new daughter DNA, okay? Whereas a semi-conservative model shows that you have the old strands of DNA acting as templates, so there's our template, and then the daughter strand. So when you have 2 daughter DNA molecules, half is old and the other half is new. Okay, so half is old, half is new, semi-conservative. And then when this DNA molecule goes through DNA replication, each strand is going to act as a template. This is the original old strand, which is a navy blue, okay? And then here's the new DNA strand. And then this would be like, let's say this one right over here is act as a template, and then this strand is going to act as a new DNA strand. Okay, so semi-conservative. And then there's dispersive, and to me this is like the most bizarre one, okay? Where it says like you have the original strands of DNA, but then in the two daughter strands you have pieces and segments of old and new. Okay, so dispersive. Every time DNA is being made, you get pieces of old and new. So Meselson and Stahl, they were the two scientists that prove that DNA replicates semi-conservatively. They basically were dealing with two different kinds of nitrogen. Remember the nitrogenous bases of DNA containing nitrogen. They made in the laboratory, they used bacterial DNA. They allowed for bacteria to grow and divide and utilize heavy nitrogen. Heavy nitrogen is nitrogen 15. And then later on, they allowed for the DNA molecules to contain nitrogen 14. Okay, but they started off with nitrogen 15, which is heavy nitrogen. Okay, so they had bacteria cultured, meaning like they allow for the bacteria to grow, make many, trillions of bacteria in this growth medium. And the only nitrogen that was available to the bacteria to make more bacteria and to make more DNA was nitrogen 15. And then they transferred the bacteria to a nitrogen 14 containing medium. So this guy, this medium had nitrogen 14, which was light. And then they tested the DNA. What kind of DNA molecules are in there? Did they have nitrogen 15? If it was conservative, you would have two strands of DNA that was nitrogen 15, and then the two strands of DNA in the new DNA to be nitrogen 14. Okay. What they did was they did centrifugation. To centrifuge something means to like put molecules or put the sample, whatever you're sampling, put the sample in a little tube and then put it in a machine that spins very, very fast. And when it spins very, very fast, all the heavy stuff goes to the bottom, and then all the light stuff will stay further at the top. I don't know if you guys have ever been to Disneyland before, or any kind of carnival where they have a ride that spins, right? When you're spinning around, but because of the gravitational pull, everything is being pulled to the end, right? To the edges. So centrifuge does that. It like pulls everything to the end, and the heavier it is, it gets pulled further to the ends, to the edges, I should say. Okay, so nitrogen 15 is heavier, and it will be pulled to the bottom, or pulled to the edges. Okay, so in their experiment, actually, let me just. Okay, so like I said before, the starting bacteria, for many generations, they only had available to them nitrogen 15. So the two strands of DNA, so I'm going to just draw the two strands of DNA. The nitrogenous bases had nitrogen 15, heavy nitrogen 15. So what I mean by nitrogen 15 in this diagram, what it means is that the two strands of DNA are both made with nitrogen 15. Okay? So in the first round, this is the prediction part. If it was conservative, okay, yeah. If it was conservative, starting, this is the tube. This is the tube for the centrifuge, okay? Starting conservative model is that you're going to have just nitrogen 15. The two strands are going to be nitrogen 15, and then the new strand of DNA, when the bacteria is transferred to nitrogen 14 medium, they're going to be using the nitrogen 14 to make new molecules of DNA, and it's going to be completely made out of nitrogen 14. So remember, the conservative model states that the old strand of DNA is conserved, and then they act as templates for new strands of DNA, but then the old strands come back together. That's that one right there. And then that's the daughter DNA, the new DNA, which contains only nitrogen 14. Whereas with semi-conservative idea is that after DNA is allowed to replicate, you have half nitrogen 15, half nitrogen 14, right? So originally we started off with nitrogen 15, but then if you allow for the DNA to replicate, each strand is going to act as a template. Nitrogen 15. Oh, this is so messy. Nitrogen 15. And then the nitrogen 15 strands act as a template for nitrogen 14 because they transferred the bacteria to nitrogen 14 containing medium. Can you guys see that? I hope so. Okay. So basically, guys, half of the strand is made with heavy nitrogen 15, which is the original, and then the 2 new strands are made with nitrogen 14. So that's why when you centrifuge the DNA, it's in the middle. It's in between nitrogen 15 line and nitrogen 14. Okay? So this line right here represents DNA strands that are completely 100% nitrogen 15. This one shows 100% the 2 strands of DNA made with nitrogen 14. But then this line, do you guys see how it's like in between? It's in between those two. It's not as heavy as nitrogen 15, 100% nitrogen 15. It's not as light as 100% nitrogen 14. So you see it in the middle. Okay? And then the dispersive, it's going to be in the middle too, because remember, it's all like pieces and bits and pieces of the old and the new. So nitrogen 15 and 14. Okay? So they did the experiment where they allowed for the DNA to replicate another round, okay? If it was, if it was conservative, you would still end up with the original nitrogen 15, and then the daughter subsequent round of replication, all the new DNA is going to have just nitrogen 14. But for semi-conservative, this is the first round of DNA being replicated. But then if you let DNA replicate again, with just nitrogen 14, you're going to end up with just nitrogen 14 band, okay? Like, for example, see up here, right? This is nitrogen 15, nitrogen 14, nitrogen 14, nitrogen 15. And then they split up, and then the nitrogen 15 acts as, I'll just try my best to draw this, as a template. That nitrogen 14 is going to act as a template. That nitrogen 14 is going to act as a template, and then that nitrogen 15 is going to act as a template. So you're going to end up with, this is nitrogen 14. This is going to get too crowded, I believe, okay? But basically, guys, this one and then this one is all pure nitrogen 14, whereas the original nitrogen 15s are on the outside. They're going to be half nitrogen 15, half nitrogen 14. And that's why you see these 2 bands. And then dispersive is just going to, stay half nitrogen 15, nitrogen 14. And then, because it's always dispersive, it's just going to contain bits and pieces of the old continuously. Okay? And this is just a microscopic, X-ray, not microscopic, X-ray photo of what they got. Okay? Which proved that it's semi-conservative. Okay? There's a picture of mesylsilinsol. Okay, so I think, yes, now let's do some practice questions, okay? So, this is just a picture of mesylsilinsol back there having some beers. Sometimes science people, even though they work really hard and are kind of nerdy, they can also be fun people, I guess. All right, let me go ahead and move my face to the side here. Tobacco mosaic virus have RNA rather than DNA. Chapter 19 is about viruses, and that's one that I didn't put on our calendar to go over, but we might go over viruses after the AP exam, because I know that some of you guys are interested in learning about viruses, especially right now with the pandemic. In a hypothetical situation where RNA from tobacco mosaic virus is mixed with proteins from a related DNA virus, the result could be a hybrid virus. So you guys remember viruses, especially like bacteriophages and tobacco mosaic viruses, are very simple structured things. I'm not going to call them organisms because some people don't believe that they're alive. They only have genetic material and proteins. They only are made up of 2 things: genetic material and protein. So what this question is setting up for you is that, you know, we're making a synthetic virus. It's mixed with its own DNA, but then proteins from a different virus that has DNA as its genome, okay? So it's going to be a hybrid. The hybrid is going to have proteins from the DNA virus, but then the genetic material will be RNA, will remain as RNA. If the virus were to infect a cell and reproduce, what would the resulting offspring viruses be like? A, tobacco mosaic virus, the related DNA virus, a hybrid, tobacco mosaic virus RNA and protein from the DNA virus, a hybrid tobacco mosaic virus protein and nucleic acid from the DNA virus, or E, a virus with a double helix made up of one strand of DNA complementary to the strand of RNA surrounded by viral protein. It's asking about the offspring. The offspring. What are the offspring going to be like? Well, first of all, remember offspring get get genetic information from parents, right? What's the genetic information that's being passed down? Is it the DNA from the DNA virus that's being passed down? No, the only thing that's part of the hybrid parent is that it's the protein from the DNA virus. But it's the RNA that is the genetic material. And that RNA came from the tobacco mosaic virus. So the answer is actually A. Even though the outside, let's just draw a bacteriophage just to keep it simple, okay? So the outside is DNA viral proteins, but then on the inside is RNA from the tobacco mosaic virus. And when it replicates, it's going to be using the RNA from the tobacco mosaic virus, which has genes for the tobacco mosaic virus. So it's going to produce offspring using that genome from the tobacco mosaic virus. It's therefore going to be all tobacco mosaic virus, okay? Okay, cysteine makes up 38% of the nucleotides in a sample of DNA from an organism. What percent of the nucleotides in the sample will be thymine? Okay, so as you guys know, DNA is made up of adenine, thymines, and guanines and cytosines. Basically what this question is setting up for you is that in a genome of an organism, 38% is cytosine. Can you tell us what percent is going to be thymine? Okay, this question might seem like, Ms. Shim, I don't have enough information to figure out how much thymine and adenine there are. But remember, guys, the amount of guanines and cytosine are equal to each other because they pair up. So if cytosine is 38%, therefore guanine is also 38%. Okay, and then you just have to add them together. What's 38 plus 38? My goodness, I can never do the math here. 76. Okay, so that means that adenine and thymine, when you add those together, they're gonna be equal to, what's 100 for, sorry you guys, I always 24. Okay, so they equal 24. So 38 guanine and cytosine is 76% total, whereas the other half of DNA is the adenine and thymine. The other half of DNA is going to be 24%. So what does that make adenine and thymine? 12 and 12. Okay, I hope you understood what I just did there. If cytosine is only 38%, it pairs up with guanine, which is also equal to 38%. The rest of the DNA molecule, not other half, but I should say the rest of the other molecule is adenines and thymines, okay? Oh, okay, so it's 12%. Sorry, I got distracted. You guys, my phone is right next to me, I got distracted. It's no wonder, like in this age of remote learning, everyone is distracted. Even teachers. In an analysis of the nucleotide composition of DNA, which of the following is true? A is equal to C? No, right? A is equal to T, right? Is the amount of A equal to G and C? No. Okay. Is A plus C equal to G plus T? Okay. Well, remember back in this previous slide, if we add, what does it say there? A plus C. If we add A plus C, is it going to be equal to T plus C? It actually is, right? Because 12 plus 38 is going to be equal to 12 plus 38. So let's go ahead and see. Maybe. What about D? A plus T is equal to G plus C? No, okay, because 24 is not going to equal to 76. So the answer is going to be C. Okay, C. Yep. Okay, so this goes back to the Mendelssohn and Stahl. Oh no, it actually doesn't, because this is asking about conservative matter. Conservative, okay? A space probe, ooh, like Mars, right? A space probe returns from a culture with a culture of microorganisms found on a distant planet. Analysis shows that it is a carbon-based life form that has DNA. That would be really, really, really amazing if that was true. You grow the cells in nitrogen-15 medium for several generations and then transfer it to nitrogen-14 medium. Which pattern in this figure would you expect if the DNA were replicated in a conservative manner? Remember, conservative means that the old DNA strand is maintained, okay? So the old one is going to have nitrogen-15. So it looks like answer choice C, D, and E could not be the answer choices at all because they don't even have nitrogen-15 available as a choice, okay? So, and then it's transferred to a nitrogen-14, and this one doesn't even have nitrogen-14. So the answer is B. Okay, that was actually simple. By the process of elimination, I came to that answer. You know what? This is just like the other one. In analyzing the number of different bases in a DNA sample, which result would be consistent with the base pair rules? A does not equal G. A plus G is equal to C plus T? Possibly, right? Because they go together. A plus T? No. You guys, this is the same question, but it's just these two are added. Okay? So B is the correct answer. Imagine the following experiment is done. Let me just see here. Okay. Bacteria are first grown for several generations in a medium containing the lighter isotope. So we're starting with this, nitrogen-14. Then switch into a medium containing nitrogen-15. The rest of the experiment is identical to Mendelssohn and Stahl, meaning like they let them, replicate over and over again in the nitrogen-15. Which of the following represents the band positions you would expect after 2 generations? Okay, so this is the control tube, and when it says control tube, it's just basically saying this is what it would look like if it had just nitrogen-14. This is the position of the DNA if it was just nitrogen-15. So this is a way of it's like a measurement tool, like a ruler. Okay, this is like a ruler. Okay, so we started off with nitrogen-15. Oh, sorry, nitrogen-14, and we know that DNA replicates semi-conservatively. So after the first round of replication, it should be a hybrid. Shouldn't it be hybrid? Half nitrogen-14, half nitrogen-15. Okay, so yeah, this is a hybrid. What is this one? I think that one's like down here. I don't know what that. Oh, maybe that could. No, I was gonna say dispersive, but no. So we're gonna cross out B. We're gonna cross out C because I do not see a hybrid. So do you guys see how like when you're answering these questions, you can start eliminating answers because you know like part of the answer, you know that it should be hybrid, but I don't see a hybrid there. I see D, that's a hybrid, so that's okay, and I do see a hybrid. So the answer is either A, D, or E, but then it's allowed to replicate in nitrogen-15. So the next generations of DNA should be nitrogen-15, which is heavier. It's heavier, right? So I don't see a heavy band here, so we can cross out A. I do not see a heavy band in D, so we can cross out D. I do see a heavy band with E, so E is the correct answer. Okay? And I think that's it. Yeah, that's it. Okay, this is where we began, so we came back to where we began. Okay, so this was a lot faster, shorter, I should say, video. I hope that it was helpful and informative, as Mr. Anderson says. Okay, guys, see you later. Bye.